This is a real event. For centuries, it has been a whisper in the woods, a fleeting shadow in the periphery of our orderly world, a story told around campfires to curdle the blood and quicken the heart. The creature has 1,000 names: Sasquatch, Yeti, skunk ape, the wild man, but it is known to most by a single, resonant title: Bigfoot. For generations, the debate has been a simple binary. Either the creature is a myth, a fabrication born of folklore and misidentification, or it is a relict hominid, a great ape that somehow survived extinction to roam the vast, unmapped wildernesses of North America. The search for an answer was a search for a body, or at least a bone. But in the 21st century, a new and more powerful tool emerged, one that promised to cut through the grainy films and plaster cast footprints to the very code of life itself. DNA. The expectation was clear. If the samples were genuine, the genetic sequence would prove, once and for all, that Bigfoot was an unclassified species of ape. But when the results of the most extensive genetic study ever undertaken on the subject were finally compiled, the conclusion was not a triumphant confirmation. It was something else entirely. Something far more complex and infinitely more disturbing. The data did not point to an ape. It pointed back, impossibly, at us. The modern saga of Bigfoot began not with a sighting, but with a footprint. In August 1958, a bulldozer operator named Jerry Crew discovered a series of enormous, human-like tracks pressed into the mud around his construction site in Bluff Creek, California. The prints were 16 inches long, eerily detailed, and belonged to no known animal. Crew, unnerved, made plaster casts. When the local paper, the Humboldt Times, ran the story, columnist Andrew Genzoli coined the term Bigfoot to describe the mysterious creature that made them. The name stuck, and a North American legend was cemented in the public consciousness. For the next decade, sightings and track finds proliferated, but they remained anecdotal, easily dismissed as hoaxes or mistakes by bears walking on their hind legs. That all changed on October 20th, 1967. In the same Bluff Creek area, two former rodeo riders and aspiring Bigfoot hunters, Roger Patterson and Bob Gimlin, were riding on horseback along the riverbed. As they rounded a bend, they came upon a sight that would become one of the most analyzed and debated pieces of film in history. There, by the creek, was a large, hairy, bipedal creature. It turned to look at them, its expression a mixture of what Patterson later described as contempt and disgust, before walking away with a smooth, powerful, compliant gait unlike any human or costumed actor could seemingly replicate. Patterson's rented 16mm camera shook violently as he scrambled to capture the moment. The resulting 60 seconds of footage, now known as the Patterson-Gimlin film, became the icon of the phenomenon. Skeptics immediately cried foul, claiming it was a man in a gorilla suit. Yet decades of analysis by cinematographers, biomechanical experts, and special effects artists have failed to deliver a consensus. Many who have studied the film point to anatomical details that seem impossible to fake in 1967. The visible muscle flexion in the thighs and back, the non-human proportions of the limbs, the unique mid-tarsal break in the foot as it walks. The creature affectionately nicknamed Patty moves with a fluid power that has never been convincingly duplicated. The film remains a tantalizing enigma, a moving portrait of something that should not exist. In the years that followed, the evidence continued to mount, forming a vast and perplexing mosaic. Thousands of eyewitness reports flooded in from every corner of the continent, from the dense forests of the Pacific Northwest to the muggy swamps of Florida. These were not just fleeting glimpses. Witnesses described towering, powerfully built hominids covered in dark hair with deep-set eyes, a pronounced brow ridge, and a conical head. They spoke of the creature's incredible speed and stealth, their ability to move through dense undergrowth without a sound and to vanish as if into thin air. Beyond sightings, physical evidence accumulated. Countless footprint casts were collected displaying dermal ridges, the fingerprints of the feet in patterns inconsistent with any known primate, including humans. Nests or ground beds made of woven branches were found in remote areas. Hair samples, when analyzed, were often dismissed as belonging to bears, elk, or other known animals, but a stubborn percentage remained unidentified, labeled by labs as unknown primate or anomalous hominid. Perhaps most unnerving were the audio recordings. The Sierra sounds recorded in the early 1970s in the Sierra Nevada mountains captured a series of complex whoops, howls, and what sounded eerily like a sophisticated form of speech, a chatter full of syntax and inflection that defy easy classification. The sounds were analyzed by linguists and naval intelligence sound experts who concluded they were produced by a biological entity with a vocal range and lung capacity far exceeding that of a human. This was the state of affairs for 50 years, a mountain of compelling but ultimately circumstantial evidence. Every footprint could be a hoax. Every sighting could be a mistake. Every sound could be a known animal, distorted by distance and terrain. The scientific community for the most part remained aloof, viewing the subject as fringe pseudoscience. What was needed was a smoking gun, an irrefutable piece of biological proof. What was needed was the genome. Enter Dr. Melba Ketchum. By the 2010s, Ketchum was a respected professional with a 27-year career in veterinary medicine and genetics. She ran her own company, DNA Diagnostics, which specialized in animal forensics and gene sequencing, often working with law enforcement on animal-related cases. She was not a Bigfoot enthusiast or a wide-eyed believer. She was a scientist, approached by researchers in the field who believed they had collected legitimate samples and wanted them subjected to the most rigorous testing modern science could offer. Intrigued by the challenge, Ketchum agreed. In 2007, she launched a comprehensive, 5-year study that would come to be known as the Sasquatch Genome Project. The project was unprecedented in its scope. Ketchum and her team amassed 111 samples of alleged Sasquatch DNA, collected from 34 different sites across 14 states and 1 Canadian province. This was not just a handful of dubious hairs found on a branch. The collection included tissue, blood, saliva, and other biological materials. The chain of custody for many samples was meticulously documented. One sample was a piece of flesh with attached hair, obtained from a creature that was reportedly shot and killed in the Sierra Nevada mountains in 2008, the so-called Sierra Kills event. Another was a saliva sample scraped from the side of a home in rural Georgia, where a family had been leaving out food for a group of the creatures. One particularly compelling sample was blood and tissue scraped from beneath the bumper of a vehicle that had struck one of the creatures as it ran across a road at night. The team, comprised of specialists in genetics, forensics, and pathology, set to work with a clear hypothesis. If the creature was real, it would most likely be a previously unknown species of great ape. Its DNA would be distinct from humans, chimpanzees, gorillas, and orangutans. The methodology was robust. They employed a battery of advanced genetic techniques, including whole genome sequencing, the same technology used to map the human genome. They extracted DNA, amplified it using polymerase chain reaction, and sequenced it. To guard against contamination, a constant plague in forensic genetics, samples were processed in multiple independent laboratories, and the results were compared. The first results to come back were from the mitochondrial DNA, or mtDNA. This type of DNA is passed down exclusively from the mother and is often used to trace maternal lineage. The team sequenced the mtDNA from 20 different samples. The result was a shock. In every single case, the mtDNA was unambiguously human. Specifically, it was modern Homo sapiens. This was the first major hurdle. The most obvious and scientifically conservative explanation was contamination. Perhaps a researcher had coughed over a sample, or human cells had been introduced during collection or processing. It was a plausible explanation, and one the team took seriously. But it didn't quite fit. The contamination would have had to be systematic, occurring across dozens of samples from different locations, collected by different people over many years, and processed in different labs. Furthermore, when labs test for contamination, they typically find a mix of DNA. Here, they were getting pure, clean sequences of human mtDNA, as if it were the primary component. A second, more disturbing possibility began to form. What if it wasn't contamination? What if the mothers, somewhere in the deep past, were human? This bizarre finding, however, paled in comparison to what they found next. The team moved on to the nuclear DNA, or new DNA. This is the DNA found in the cell's nucleus, inherited from both parents, and it contains the vast majority of an organism's genetic information, the complete blueprint for what it is. After successfully sequencing 3 whole Sasquatch genomes from the most promising samples, the true anomaly revealed itself. The nuclear DNA was a mosaic. Part of it was clearly human, consistent with the mtDNA findings. But the rest of it was something else. It was primate, but it did not match any known primate species on Earth. It was not gorilla, not chimpanzee, not orangutan. It contained novel genetic sequences that had never been documented before. The genome was a chimera, a hybrid of 2 distinct lineages. One was modern human, the other was an unknown, unidentified primate. Ketchum and her team checked and rechecked their findings. They ran the sequences through GenBank, the massive international database of all known genetic sequences maintained by the National Institutes of Health. Time and again, the results came back the same. A significant portion of the genome was no match. This wasn't a case of a few anomalous genes. This was a structurally unique genome, part human, part unknown. Based on their analysis of the genetic data, Ketchum's team reached a staggering conclusion. Sasquatch was not an ape, it was a hybrid species, the result of a cross between female Homo sapiens and males of an unknown hominin primate. Based on molecular clock dating of the sequences, they proposed that this hybridization event occurred relatively recently in evolutionary terms, likely around 15,000 years ago. The implications were world-altering. This was not an ancient, ape-like ancestor like Australopithecus. This was a contemporary species, walking the earth alongside us, that was part human. It meant that our own species had interbred with another intelligent, bipedal primate in the not-so-distant past, creating a viable, breeding population that has survived in secret to the present day. The wild man legend was not just a beast of the forest. He was our cousin, a very, very close cousin. Armed with what she believed was one of the most significant discoveries in human history, Ketchum prepared her manuscript for publication. She submitted it to the most prestigious scientific journals in the world, including Nature and Science. The response was swift and brutal. The paper was rejected without peer review. The gatekeepers of mainstream science would not even look at it. The conclusion was deemed too extraordinary. The subject matter too taboo. The human mDNA was seen as definitive proof of contamination, and the novel new DNA was dismissed as likely being the result of sequencing errors from a jumble of bacterial, fungal, and human DNA. The scientific establishment, in essence, declared the data invalid because the conclusion it led to was impossible. Frustrated but undeterred, Ketchum took a controversial and professionally risky step. She acquired an existing, little-known science journal called the De Novo Scientific Journal and, after what she stated was a rigorous peer review process by a panel of anonymous experts, published her own paper in February 2013. The paper was titled, Novel North American Hominins, Next Generation Sequencing of 3 Whole Genomes and Associated Studies. The reaction from the broader scientific and media world was a firestorm of ridicule and condemnation. Ketchum was branded a crank, a fraud, and a laughingstock. Her decision to publish in her own journal was seen as a fatal blow to her credibility, a classic sign of someone peddling pseudoscience. The Sasquatch Genome Project was written off as a cautionary tale of bad science, confirmation bias, and wishful thinking. The door that had been briefly cracked open on the mystery was slammed shut, bolted, and barricaded. The official story became that the Ketchum study was a failure, its data worthless. The mystery of Bigfoot remained unsolved. But the question lingered for those who looked closer. What if the data wasn't worthless? The sequences were real. The 3 billion base pairs of the 3 sequenced genomes were uploaded and are, to this day, publicly available on GenBank. Dismissing the conclusion is one thing. Dismissing the raw data is another. If it was all just a messy compilation of bear, human, and bacterial DNA, other independent geneticists could easily analyze the public data and prove it. Yet, few have. The subject remains so toxic that serious academics will not touch it. The data sits there, a digital ghost in the machine, largely ignored. This is where the true mystery begins, a mystery far deeper than whether a giant ape roams the woods. If we accept, for a moment, the possibility that Ketchum's team found something, even if their interpretation was flawed, several chilling theories emerge from the shadows. The first is Ketchum's own theory, the human hybrid hypothesis. If true, it redefines what it means to be human. We are not the sole inheritors of this planet's intelligence. A sister species, born of our own lineage, lives in the margins, a wild reflection of ourselves. This explains their intelligence, their elusiveness, their seemingly human-like behaviors. They are not animals to be hunted or captured. They are people to be understood. The legal, ethical, and moral implications are staggering. Would we have rights? Would they be a protected species or a protected people? The very existence of a human hybrid would shatter all religious and philosophical foundations. Perhaps the reason the discovery was so violently rejected was not because it was bad science, but because humanity is simply not ready for the truth. It is easier to believe in a monster than it is to accept a relative. The second theory is a darker variation of the first. It suggests that the unknown primate portion of the genome is not from some gentle, relict hominid. It is something else. Throughout history, legends have spoken of more than just wild men. They have spoken of giants, of cannibals, of monstrous entities that prey on humans. What if Sasquatch is not a shy, gentle giant, but a remnant of a predatory hominid line that once hunted our ancestors? What if the human DNA within them is the result not of interbreeding, but of something far more sinister? Genetic assimilation through consumption, or a parasitic form of horizontal gene transfer once thought impossible in complex animals. This casts their elusiveness in a new light. They avoid us not out of fear, but because they are patient hunters, and they know that full-scale discovery would mean war. A third theory abandons biology altogether and steps into the realm of the preternatural. Many Native American traditions do not view Sasquatch as a physical creature of flesh and blood. They see it as a spiritual entity, a guardian of the wilderness, a being that can shift between the physical and spiritual planes. This is why it can disappear without a trace, why it can move silently, why it seems to possess an intelligence that borders on telepathy. In this context, the DNA results are a red herring, a biological paradox because the creature is not entirely biological. The human DNA could be a spiritual echo, a reflection of the creature's deep connection to the land and to humanity. The unknown DNA could be something not of this world, a genetic signature that our science, based entirely on the physical realm, can never hope to classify. This theory suggests that trying to capture or sequence a Sasquatch is like trying to bottle a ghost. The results will always be confusing, contradictory, and ultimately meaningless, because the tools of our science are insufficient for the task. Finally, there is the most pragmatic and perhaps most terrifying theory, the cover-up. Let us assume Ketchum's data was 100% correct. A small, unaccredited lab in rural Texas had just made the single greatest anthropological and biological discovery in history. The news would not just be world-changing. It would be world-destabilizing. Consider the consequences. The logging industry in the Pacific Northwest, a multi-billion-dollar enterprise, will be shut down overnight to protect the habitat of a newly discovered, sentient, human-related species. A land rights battle of epic proportions would ensue. Religious doctrines about the special creation of man would be thrown into chaos. It would be, in short, a global crisis. Is it not conceivable that certain powerful interests, governmental, corporate, or scientific, decided that this discovery must be suppressed? It would be far easier to orchestrate a campaign of ridicule, to discredit the source, to bury the data under an avalanche of academic scorn, than to deal with the fallout of the truth. In this scenario, Melba Ketchum was not a bad scientist. She was simply the first to break a code of silence that has been enforced for decades. The scientific establishment did not reject her findings. They were instructed to. The project is now a decade in the past. Melba Ketchum has faded from the public eye. The world has moved on. Bigfoot has returned to the realm of campfire stories and blurry YouTube videos. But the mystery she uncovered has not gone away. It festers. The genetic data remains online, a silent testament to a question that was asked but never truly answered. And out in the deep woods, the phenomenon continues. But some observers and longtime researchers report that something is changing. The vocalizations are becoming more frequent, more complex. The sightings, once confined to the deepest wilderness, are creeping closer to the edges of civilization. Reports are emerging of the creatures observing human dwellings for extended periods, not with aggression, but with a quiet, unnerving curiosity. They seem to be studying us. A recent audio file, captured by an automated recording station in a remote Washington valley, is particularly unsettling. 4 hours, it records only the sound of the wind and the distant call of an owl. Then, a new sound begins. It is the familiar, powerful whoop of a Sasquatch, but it is patterned, rhythmic. It is followed by a series of guttural clicks and a low, resonant chatter, the language of the Sierra sounds. But woven within it is something new. A sequence of tones, modulated with an intelligence that is impossible to dismiss. It is an attempt at mimicry. The creature is trying to replicate a sound it has heard from our world. It isn't a word, not yet. It is a sequence of notes that sounds disturbingly like a distorted, three-tone cell phone ringtone. A sound that means only one thing, an incoming call. The question is no longer just what these creatures are, or where they came from. The question now, as they mimic the very technology we use to communicate, is what are they trying to say? And are they finally getting ready to make the call themselves?